The paper what I'm going to cover is the very primitive research about isolation and identification of cancer stem cell in human brain tumors. Nowadays, heterogeneity and diversity of cells in cancer tissue is well known to researchers, but even before decades ago, understanding about tumor environment was not that clear. This paper suggested early ideas for presence of essential subpopulation in brain tumor, which is multipotency, capacity for multilineage differentiation, and sphere-forming ability. Tumor-associated environment, including fibroblast, endothelial, tumor-associated blood vessels, and other surrounding components, helps cancer cells to maintain tumor-friendly environment or metastasized to other region. Among this population, essential subpopulation named cancer stem cell play a key role for heterogeneity of tumor tissue. Due to its importance, efforts to identify and isolate those populations have been continued. As you can see in the cartoon, various approaches have been proposed, and each method focuses on certain properties of the cancer stem cells. For example, ALDH method is application of high ALDH expression of in progenitor or stem cells. Identification of brain tumor stem cell, however, changed paradigm of brain tumor treatment. As we can see in the picture, conventional radiation therapy has limitation due to the radiation resistance of cancer stem cell or CD133 positive cells. But pre-treatment of cell growth factor like bone morphogenetic proteins induces loss of stemness of CD133 positive cells and make the following treatment more effective. Back to the paper. Now let's take a closer look at cancer stem cell isolation. In this paper, they used various types of primary cancer cells. First figure showed there were various cancer cells that forming spheres in vitro. From the left column, medulloblastoma, pilocytic astrocytoma, ependymoma, and ganglioglioma cells are shown. First row shows in vitro image of each primary cancer cells. Second row is tumor sphere image of nestin and CD133 stained. Third row shows differentiated cells, also nestin and CD133 stained. Next, they showed sphere-forming cells exhibits increased self-renewal potential compared with control. Medulloblastoma cells, which showed relatively high second sphere-forming capacity, also showed enhanced self-renewal potential at limiting dilution assay. Picture C, D, E shows secondary tumor spheres still expressing nestin and CD133. They also observed that tumor sphere cells exhibit increased proliferation consistent with histopathologic mitotic indices. Tumor stem cells from dissociated medulloblastoma spheres showed excessive degree of cell proliferation than pilocytic astrocytoma or normal neurosphere cells. They also checked tumor sphere cells can differentiate into other immunophenotype and confirmed it with various markers. Tumor sphere cell marker CD133 and nestin was no longer highly expressed by the cells once they differentiated into certain phenotype. Instead, differentiated cells showed altered marker expression pattern. In case of pilocytic astrocytoma, differentiated cells CD133 and nestin expression dramatically decreased, and GFAP expression increased abruptly. Medulloblastoma spear cells showed same change in expression pattern too. Since they found CD133 positive cells have distinct properties compared to others, they identified the general feature of CD133 positive cells. Similar to previous findings, CD133 positive cells showed marked stem cell activity. After they sorted the CD133 positive primary medulloblastoma cells with flow cytometry, tumor spear formation and proliferation capacity was checked. And they found there's a huge difference between CD133 positive and CD133 negative cells. Difference among normal neural stem cells, CD133 positive cells and CD133 negative cells is also investigated. Huge difference between normal stem cells and cancer cells was excessive expression of beta-tubulin in cancer cells. Authors suggested that these differences originated by genetic abnormality based on their finding that tumor spear cells showed abnormal karyotype, including loss of chromosome 10, 16, and gain of chromosome 18. This paper provided general and primary view for brain tumor stem cells, cited by many other researchers numerous times. Although the concept of cancer stem cell is still controversial in some cases, it is clear that certain populations in tumor cells are responsible for various stem-like properties of the tumor tissue.